Then it will be broken down into what we call fatty acid and glycerol. Then this will also be metabolized in your body through different ways. All the ones you don't now need in your body, after you have assimilated the ones you need, all the ones you don't need must be getting rid of. And you get rid of them through what? Excretion. Abi? So you started from nutrition, you pass some things into your body. Then after passing it, you study transport system that helps you to carry those things to everywhere they are needed. Then after that, you make use of the ones you need by producing energy. Then you study respiration. Then after that, you get rid of those ones you don't need. Then you study excretion. Then when you are done with that part, the muscular part of your body that helps you to carry out the transportation, excretion, respiration, you also have to study it. Then that's when you start talking about the skeletal system. So all the activities of your body will now be carried out by some selected muscles, bones, joints. Then that will not only work in isolation, it works with your nervous system. So your nerves, your hormones, they tell your body when you are hungry, they tell your body when you are to digest, they tell your body when you are to move and coordinate other activities. And that's why we call that hormonal coordination. Then when you are through with skeletal system, you have done hormonal and nervous coordination. Is there any other coordination again for the systems? That's all. You talk about reproductive system, because reproductive system is your existence. You have to continue. You are carrying out a function, then you need to give rise to another organism that will carry on your existence. Then that is reproductive system. So we'll do that. And when we are doing this, we'll be aligning it with plants too. So we do for animals, we do for plants, and as much as possible, let's try to get the best out of it. It's not so hard. Then when we are done with all our own self, when we are done, then it now comes to our relationships. And that is where you start studying ecology. What activities, how we affect one another, what you do, how it affects me, what I do, how it affects plants, what plant does, how it affects me. Then you start talking about relationships. And that is when you start studying everything about ecology, where we live and all of those things. Then where plants live too, because at this level, everything you talk about goes for both plants and animals. But when you get to university and you want to study a course, then there will be a particular section you focus on, either plants, either animals, or the general thing. Then when you leave that part, it is good to also learn the basics of your body, the system, what makes up your body. We have talked about transportation. Then you have to look at it deep within the cell. Then that's when you start talking about genetics. What makes you carry out all of these functions? Why are you kind of different from every other person? Then you start talking about genetics. Then one important aspect that JAMB can't do without is genetics, adaptation, and evolution. So evolution is now the question of biology. How did we get here? That's evolution. And we know that biology is defined as the study. And we know that biology is defined as the study of life. As it is, they used to ask. So when you're now talking about biology, I just want to pick all the important points and not deeper that you will need. I will also let you know. So for the living organisms, we all know the general characteristics. Mr. Ninja Abi, and that's what I just talked about. Now, the most important part is all of us are made up of something that we call cell. Abi, I only have 30 minutes to cover all this part. So for cell, you need to know the first thing is all of you must know what a cell is. Abi, and you all know by now. Then we know when we're talking about organization of life, we say cell we aggregate to form tissues. Abi, and tissue we aggregate to form organs. And organs we aggregate to form system, or what you call the organ system. And the system we aggregate to form the whole organism. So this is the basics of biology that makes you. And a whole lot you have to know about cell, we'll talk about it. So when you have similar cells performing similar functions, then you say you have tissues. Then which means that if you have similar tissues performing similar functions, you have organs. When you have similar system performing similar functions, then you have an organism. Now, looking at what we have here, let me just brief this. I can have what we call the RBC. RBC is called the red blood cell. I can have white blood cell, WBC. Abi? Now, when they all come together, they form a tissue. And that tissue is what we call blood. So you can see. Now, when this blood come together to perform a specific function with other muscles and all of those things, you form the hearts. Abi? Now, these hearts will now form a system of transport that you have from head to leg everywhere. Then you now have what we call the cardiovascular system. So the cardiovascular system, also known as your transport system, is what you now deal with here. Then that makes part of your body. But there are several systems that make up the organism. Digestive system, respiratory, and so on. So now when you're talking about cell, there's something we all know. Is there anybody that does not know this? We call this cellular organization. Cellular organization. Now, when you look at cell, we have the animal cell that you all know, and we have the plant cell, right? Now, the basic unit is cell, and cell can be unicellular. It can also be multicellular. Unicellular in the sense that there are some organisms that are made up of just only one cell. All of us already know these cells. Now, if I may ask, is there anybody that does not know the function of nucleus? And don't be shy. It's better you say yes, you don't know. I mean, no, you don't know. So that we explain it. Is there anybody that does not know the function of nucleus in this place? Is there anybody that does not know the function of endoplasmic reticulum? Is there anybody that does not know the function of nucleolus? Where is nucleolus found? In the nucleus. Is there anybody that does not know the function of smooth endoplasmic reticulum and rough endoplasmic reticulum? Yeah? Now, for the smooth endoplasmic reticulum, so to all of you, there's a plant cell and there's an animal cell. So in the plant cell and animal cell, you have a center. That center is called the nucleus. Then you have a cell that produces ribosome. That is called your nucleolus. This one contains some kind of thread-like materials. Those are called chromatins. Chromatins. Then this is called your nucleolus. Why? Now there's an opening here that forms a fold. Now, on top of this guy, you can have some things that have been synthesized here. What did I say nucleolus produce? Ribosomes. So ribosomes come out of the cell. They can attach to something. That structure they attach to is called rough endoplasmic reticulum. Now, there's another endoplasmic reticulum that does not have, so to put it, it does not have the ribosome on it. Then this one is called smooth endoplasmic reticulum. Now, when you're talking about lipid synthesis, production of lipids, then you have what we call the smooth endoplasmic reticulum. But production of proteins, majorly, production of proteins and modification of proteins, then you have the rough endoplasmic reticulum. Is that taken? But when you are talking about production of lipids, you are talking about smooth endoplasmic reticulum. Now, this nucleus has a point, we call it the nuclear membrane, Abi, the nuclear membrane. Inside the cell, you now have some other organelles, like the one that forms a matrix like this. When you see a structure like this, what structure is this? Mitochondria. And you all should know the function of mitochondria. You also have another one that contains some structures that are linked together by a thread kind of thing. Then that is called chloroplast, but you should not see it here. Now, there's another one that looks like sausage with some vesicles on it. That is called what? Golgi apparatus, because they can use the structure to ask you a question. So I'm not supposed to waste time here. So that is called Golgi apparatus. Now, you have some of these things too inside the plant cell. But inside the plant cell, you have a big vacuole. Some animals too have vacuole, but it's very small. Like amoeba. Amoeba has vacuole. They have food vacuole, they have contractile vacuole. You remember that? So then there's another structure. You have what we call the lysosome. Abi, you should know the function of lysosome. Which other one? You remember, this is called the cell membrane or the plasmalemma. These are not experts, I should be wasting time. So we call that the cell membrane or plasmalemma. Now, what makes up the cell membrane? What are the things that make up the cell membrane? That's lipids, proteins, and carbohydrates. So when they ask you in JAM that what are the major components of the cell membrane, it is lipid and proteins. Is that taken? Now, the lipids are the major components, and that lipid at your level might still be called only phospholipid. Phospholipid. And what is the function of the cell membrane? I expect all of you to know all these ones and digest it. I'm just trying to remind you. So, cell membrane controls what comes into the cell and what goes out of the cell. Abi? So, that is the function of cell membrane. So, cell membrane is said to be selectively permeable. So, you must remember that, because it controls the things that come in. It's not everything that can go into the cell, and it's not everything that comes out of the cell. So it controls what goes into the cell and what comes out of the cell. So you said it is selectively permeable. But don't forget, cell wall. Cell wall is usually found in plants, Abi. So cell wall is freely permeable. That one allows anything to come inside. So are you there? What's the function of Golgi apparatus? Now, when you want to, the proteins produced by this one will be carried in vesicles. Those proteins will be carried and they will go to the Golgi apparatus. Golgi apparatus will now have necessary carbohydrates, all the reformation and everything. So it will transform the protein to a useful one. Then it will now send it out of the cell for use. So that is the function. So it transports materials out of the cell. It's also involved in production of lysozymes too. Is that taken? So in production of lysozymes. And what is the function of lysozyme? So dead cells, anything that comes into your body that your system does not like, lysozyme can break it down. So lysozyme contain what we call hydrolytic enzyme. Hydrolytic enzymes. So the hydrolytic enzymes or digestive enzymes, they help you to break down things that your body do not want. So don't forget all of these things. Now, what are the differences between plants and animal cells? Which one has centriole? Animal cells. Which one have cell wall? Plant cells. So all of these things. What is common to them? They all have cell membrane, Abi. They have nucleus. And there's another thing you should not forget. When you're talking about cell based on origin, we have what we call prokaryotic cell and eukaryotic cell. And you all know the difference now. So for the sake of those that do not know, this one lack true nucleus, no true nucleus. That's one of the things you need to know. No true nucleus. That means this one have what? True nucleus. Is that taken? Another thing is that most of the organelles, you don't even have organelles like that. But if you have organelle at all, it is not membrane bound. No membrane bound organelle. So that's that. Why most of the organelles here are membrane bound. And that's why you have the nucleus here. The nucleus have what we call the nuclear membrane. But in prokaryotes, there's nothing like nuclear membrane. It is just what we have. What we have is what we call the nucleoid. Nucleoid. That's what you have. So I'm not comfortable with the way this thing is. So it's not allowing me to write easily. We are also saying people's chairs are too. And I need to be fast with what I'm writing. You can allow peace to probably sit at that edge. So that she can place her leg properly. Oh yeah. Now, for unicellular cells, you need to also understand something. You have heard about this, that cell can be free living. Cell can be free living. That means the cell can exist on its own, independent of another cell. An example of free living organisms are amoeba, paramecium, euglena, and the likes. Abi, why cell can exist as colony? So these are questions they can ask you. That which one exists as colony? Colony is aggregate of cells. So you have several cells carrying out functions together. Is that taken? Just single cells, but they have to depend on one another. Then that is called colony of cells. An example of common organism is what? Volvox. Then cells can exist as what? Filaments. Filaments. An example is what? Spirogyra, unbranched chain that you have in Spirogyra. So these are the most basic things. Know the functions of all of these organelles, know their impacts and everything. Then let's quickly move on to cell and its environment. So now, we said that cell membrane, you see that you build on it. That's why I had to start with it. Look at this now. Have you all written something here? We will solve the questions together on these parts and on the cell and its environment quickly. I've spent like 10 minutes here now. So now, are we okay with this? We said cell membrane controls what? What goes in and what comes out, Abi. But now, what goes in and what comes out? There are a lot about the cell membrane that we can't really talk about at this level. It's just like a house that this place now, there are blocks everywhere. There are windows, there are doors, there are things that can come out through windows, there are things that can come out through the door. You cannot pass the window, not even with this size, but you can pass through the door. So there are some things that come in here too and go out. So what brings in things and what takes them out and in what environment is what we are talking about next. Cell and its environment. So when you place cell in a particular environment, how does it behave? So let's see how it goes. Where's the duster? I might waste time. We will solve all the questions together. Then I will ask you, before we move on to the next topic, I'll summarize that too. We'll solve all the questions. I'm not sure. Is the only one duster we have? Oh yeah, let's be fast. Ojola, don't sleep now. Heritage, don't sleep. Oh yeah, someone should quickly help me look for duster. This one cannot help us. Look for duster. Be fast. So don't forget, in cell, there are some things we call microtubules or microfilaments. Microfilaments are just like the skeletons of the cell. They are the ones that hold many things together in the cell. So in case they ask you about microfilaments. Please, let's look for duster. Some cannot go. So that's just a revision and a summary of the things you have done. So in case it's your first time in class, you would have had one or two things. Mitochondria is the powerhouse of the cell. Cell wall gives rigidity to plants. Chloroplast helps in photosynthesis, the chlorophyll. Chloroplast house chlorophyll. Why chlorophyll helps in photosynthesis? Now, the chlorophyll contains what we call the stroma and the thylakoid, the grana. So when we get to nutrition, I will talk about that. Then the nucleus is the control center of the cell. It is the most obvious part of the cell when you are carrying out staining. So if they ask you that the most conspicuous part of the cell, that is the nucleus. And the nucleus carries the genetic material, that is the DNA, in form of chromatins. Then when the chromatins wrap around histone proteins, then we have the DNA. So all of those things, we have the chromosomes and all of those things deep within the cell. So you now need to know the difference between plants and animal cells. And that's all for that introductory part, except one or two things. So now for cell and its environment. So quickly, when you're talking about cell and its environment, the cell is in a particular environment. What controls what goes in and out of the cell? The cell membrane. And we said the cell membrane is made up of what? Lipids, Abby. So these lipids, proteins, and carbohydrates, they are the ones that control a lot of things about the cell. Now, lipids, we have the phospholipid. Now, when you're now talking about this cell membrane, how can things go into the cell? So let me put it this way. Things want to go into this cell through the cell membrane. How will it go into the cell? Then we call it transport. Transport. Things moving into the cell. Are you there? So just to put it that way. Now, there are two types of transport majorly. There are three types. There is passive transport, there is active transport, and there is bulk transport. But I'm going to focus on active and passive for this class. Now, there is what we call passive transport. And what is passive transport talking about? What is transport? Movement of materials in and out of the cell, Abby. So now, passive transport is the movement of materials in the normal direction. Let me put it that way. So you don't need energy in the normal direction. So where normally materials move from where it is more to where it is less, Abby. So that is the thing. So just know that when you move across concentration gradients, then you call it passive transport. And what helps to move it? No energy is needed. No energy needed. So that is passive transport. It is normal if I have too much water on one side, if I have high water here, and if I have less diluted or less water here, let me just put it that way. It is normal for water to flow from where it is more to where it is less. Abby, that is without passive transport, diffusion, and we have osmosis. Abby, is there anybody that does not know these two? Diffusion is called simple diffusion and facilitated diffusion. So if you want to move things across the membrane, is that summary I'll be giving you. So just listen and quickly write the best way you can. If there's anything, I will spend time on that. Now, for cell membrane, it controls what comes into the cell. But how will things move into the cell? It can move in by either diffusion or osmosis. But on a broad view, it moves in by what we call passive transport or active transport. I'm still on passive now. When it moves across concentration gradients, then we call it passive transport. There will be no use of energy. We will solve questions soon. Now, when it moves like gases, ions, some ions, Emmanuel, ọ̀tọ̀ nìkan tẹ́ máa máa ṣe all night. You cannot just be sitting like anywhere you go in life, no matter the threat to your life, being active in class should be your number one priority. Kò má ti kàwé for a very long time. Being active in class is your number one priority. Aside the books you will read. What we are saying now, even though you know it before, fine, but at least be active. Now, diffusion is defined as the movement of molecules from a region of what? High concentration to, so you expect that to happen on its own, Abi? So molecules move from high concentration to low concentration. Now, simple diffusion is when it just happens. Nothing is carrying those molecules. Facilitated, from the word facilitated. If I say I facilitate something, that means I made it happen. Is that taken? That's the English. So if you move molecules using some other things to carry them, then we call it facilitated diffusion. They are what we call carriers and all of those things, but we are not going to that part. So when things want to move across the membrane, they can just diffuse. Water can diffuse, gases can diffuse, oxygen can diffuse, ions can diffuse. When those things move from higher concentration to low concentration, then we call it diffusion. Is that taken? But osmosis usually focus on water. And we said that when you move water, the English I used to get students wrong, yes. You move, if you're talking about salt concentration, osmosis will be the movement of water from lower salt concentration to higher salt concentration, or water from where water is more to where water is less. From high water concentration to low water concentration, or from low salt concentration to high salt concentration. Let me use a picture to quickly show you that. So now here I have the bigger ones. Let's say these bigger ones are salts. Quickly. Then these ones are also salts. Then you have water. When you have less salt, means you have more water. Then you have less water here. Because the salts are too much, so they absorb the water. So on this side, more water, Abi, less salt, right? Now, what separates them is called a semi-permeable membrane, right? So now, you expect water to move from B to A, Abi, from left to right, because there is more salt here, and the salt will absorb the water, Abi. So now, that water moves until there is balance. So, movement of water from where water is more to where water is less, across what we call a semi-permeable membrane, is what we call what? Osmosis. Do we all get that? Now, what are the examples of semi-permeable membrane? Are common questions they can ask you in biology. What are the examples? Cellophane paper, bladder, yam that is peeled. You peel a yam, peeled yam, and so on. Anything. These are the things that can act as semi-permeable membrane. That means that we allow things to go through them or come out of them. And majorly water, so they select what goes in and what comes out. Is that taken? Now, common question they will now ask you is that when do you have diffusion? When do you have osmosis? Most times, movement of water is associated with osmosis. Is that taken? There are enough spaces at the front here. There are enough spaces here. So I don't know why there are spaces here. Some of you have to stand. There are spaces here. They are just 5 here. They can adjust. There are 5 there. Adjust. Oh yeah, oh yeah, oh yeah, oh yeah, on time. Move to the front. Adjust. They have space here. On time, on time. I have some few minutes more. So now, from what we have here, just know the second part. The second part is called active transport. You understand osmosis and diffusion now? Now, active transport is when you are going against ọmọ tó dọwọ́ bojú látàárọ̀ yẹn. Note the difference in these statements. Across, against. Against means what? Oya, oya, oya. Against means what? It's not opposite now. It means that you are going from where it is less concentrated against where it is more. Normally, you expect it to move from which is opposite. You expect it to move from high concentration to low, Abi. But now you want to let it move from low to high. Then that is what we call against concentration gradient. So if you see definition of movement of molecules or ions against concentration gradient, or from where it is lower to where it is higher, then you say it is what? Active transport. And this requires what? Energy. Requires energy. So let's move on from there. Now, what is the application of these things? A cell can be dropped inside. So let's take a cell now. We want to talk about solutions. From that experience, a cell can be dropped into a solution. So let's say this is the cell that is in the solution, then the solution, then another cell into another solution, then another cell into another solution. So quickly, what are we trying to do here? The first solution, we call it an isotonic solution. You all know what isotonic is. Same. So the concentration in the cell is the same as the concentration of the solution. So they balance out. When there is balancing like that, then we said it is isotonic. Then we have what we call hypotonic solution. And don't forget that the word focus on solution. Hypo always means less. So when there is less salt, please, when there is less salt compared to water, then we say, compared to this cell now, like the solution, concentration of salts in solution is less than concentration of salts in cell. Then you said it is what? Hypotonic. Are you getting that? This solution now has more salts. The cell too, you remember there is something called cytoplasm in the cell. The cell has its own salt concentration too. If the concentration of salts in this solution is less than the concentration of salts in the cell, then you said that solution is hypotonic to the cell. Is that taken? Is there anybody that is lost? So this one is hypertonic, Abi. Hypertonic will now mean what? The concentration of salts in the solution will be more than that in the cell. Is that taken? Now, what do you now expect from what we have said in osmosis? If there is more salts in the solution than in the cell, what do you expect? Where in what direction will water move? Is it from cell to solution or solution to cell? Because the salt in the solution is more than the salt in the cell, Abi. So in hypertonic solution, water moves from cell to solution. Is that clear? Why in hypotonic, what will happen? Water will move from where? From solution to cell. Is that taken? Why in isotonic, there will be no water movement, Abi, because they are balanced. Now, what is now the implication of this? The result of this is that when water moves out of the cell, as the cell is losing water, the cell will continue to shrink. So we call it, number one, under this place, and have the shrinkage of the cell. That shrinkage, if you're talking about plants, we usually call it plasmolysis. Abi? Plasmolysis. So when the cell shrinks, then we call it plasmolysis. Then, if it is for animals, we usually focus on red blood cell. Then we call it crenation. Crenation. Red blood cell crenates. Plants plasmolyze. Do you get the difference between the two? Shrinkage of cell is called plasmolysis if it is plants. Shrinkage of cell is called crenation if it is animal red blood cell. Look at the picture again. Do you get the concept? So when it involves plants and it shrinks because it's losing water, so it shrinks, then we call it plasmolysis. When it involves this, we call it crenation. Is that taken? Now, on this other side, because it's gaining water from the cell now, this one is losing water. This one is gaining water. This one is losing water. So yes, cell is losing water. Yes, cell is gaining water. What do you expect to happen to the cell? It becomes swollen, Abi. So the result of this hypotonic is you have a swollen plant. So if it involves plants, then we call it turgidity. Turgidity. Plasmolysis is also a form of flaccidity. Flaccidity. A plant cell can become flaccid and not plasmolyze. It can die. This one might not be able to regain its shape again, but this one might be able to regain its shape. That's the major difference. So we have turgidity, and turgidity is important in plants because it makes plants to stand very well. Then that turgidity, the vacuole, that we drew the other time, we absorb enough water, then the plant will stand. And what used to absorb that water is parenchymal cell. I'm going to get there when we get to skeletal system, or I'm talking about plant anatomy. So as we are moving on. Now, from what we have here, if it is blood, we call it hemolysis. So if the red blood cell drinks water and it bursts, then we call it hemolysis. Is that taken? So it absorbs too much water, then it breaks apart. Then we call it hemolysis. We're going to be using some terms later. But for now, is there anybody that's still confused here? Are we okay now? So in 5 minutes, let's go through the past questions on that. So yeah, bring out your past questions for biology. Then I'll quickly move on to classification. Although the classification, I saw somewhere talking about it the other day, and I think you have spent enough time on classification. But maybe just one or two things again. There's nothing to talk about then. I can move on to nutrition. Oh yeah. Everybody open. This is my honor. Oh yeah. We have not come here to play. And we want to answer everything in a very short time. Oh yeah, everybody, look at it. Are you there? Even if it is a day to your exam, whether you have finished this thing before or not, you are meant to sit down, just glance. You will just be looking at everything from beginning to the end. From beginning to the end, like a summary again. That's more like what we are doing now. It's more like revision. So, can we solve now? Which of the following characteristics is of animal cell? Absence of large vacuole. You know, plant cells also have chromosomes, Sabi. So you can't pick that. And you see why you have to be very careful. Some people might have chosen chromosome. Yes, because they start thinking about it that animal cell chromosome should be common, but plants who have chromosomes. Is that taken? That's why biology can be trickful and you still don't know. Because now they said absence of large vacuole. Some of you start thinking about it that paramecium has vacuole, kinikon has vacuole. Will it now be chromosome or vacuole? Then before you know it, that evil spirit will say choose chromosome. Then you choose chromosome and you now be expecting 100 in biology. It does not work that way. So please, biology can be very simple, but it can also be trickful. Let's just be careful and trace. Simple, trickful, careful. Number 2, all living organisms. This one is focusing on the general characteristics of all living things. Sabi? So all living organisms do what? They respire. The vacuole of plant cell is what? For plant cell, there's what we call the fluid that fills the vacuole, Abi, and it's surrounded by a membrane. What's the membrane that surrounds the vacuole of plant cell? Tonoplasts. If you don't know before, you take notes as we are solving questions now. You don't know what will come out to your own exam. They still asked about tonoplasts last year. So, what's the answer? It is not just filled with air, it is filled with liquids, Abi. That liquid is called sap, Abi, cell sap. Then it is surrounded by a membrane, and that membrane is called tonoplasts. Are you there? Number what now? Number 4. Which of them is solely responsible for respiration? Number 5. Which of the combination is common to plants and animals? We are doing this in case you have not even solved before or it's your first time. That's why we are doing this. And I'm trying to be fast about it so we can move on to the rest. C is what? Plasma membrane, chromosome, mitochondria, and lysosome. So that's common to all of them, Abi. You all know where energy is produced. Which of them is not common to both plants and animals? Chloroplast. You all know, Abi. Please be careful. You know now in this option there is vacuole and there's chloroplasts. You have to be careful, but you should know sometimes chloroplasts might not even be the answer, depending on what the question is saying. Some of you just rush at times, just pick chloroplasts. Yes, I've seen questions like that before, that chloroplast is not the answer. They were asking about why is this one not carrying out this aspect and no. Everybody was just picking chloroplast. Even some textbooks picked it too. So please, you have to know what your question is saying before you pick your answer. And I'm trying to see how. Similar cells, what? What is the function of endoplasmic reticulum? Intracellular transport of materials. You can see that protein synthesis is there now. Now, what majorly helps in protein synthesis? Ribosomes. What produces ribosomes? Nucleolus. So you should know what you are saying. Is that taken? Why we say protein is produced in endoplasmic reticulum is because of what? And the ribosome that is attached to it. Is that taken? But the major function is in intracellular transport. It will carry materials out of the nucleus and it will put it in the cell. Is that taken? All these are basic mistakes many people will make. So know the difference. Is that clear now? Which one helps in digestion and destruction of foreign bodies? Lysosome. What are the things that are present in lysosomes that helps it in digestion? Hydrolytic enzymes. Is that taken? So number 10, you already know the answer, B. Number 11, function of ribosome is what? Protein synthesis. Which of them is common to glandular white blood cells and amoeba? Cell membrane. They are just talking about animal cells. Shall you get? Then the structure labeled 5 is what? That's the Golgi body. You can see it's like that donut with a vesicle on it, Abi. When you see that thing on it, they already know that is Golgi apparatus. Which of them contains ribonucleic acid, RNA? That's number 3, Abi. That is the nucleolus. Which of the structural powerhouse? 4, Abi. So I believe to some extent you can solve the rest. 16, 17, 18. Let me look at it. Number 20, we just answered that too. You should be able to answer 21. I'm going through all of the questions to see anyone that you might not be able to answer. What's the answer to 26? Liver. What's the answer to 27? Within the cytoplasm, Abi. Please, I hope you know what protoplasm means. So everything inside the cell is called protoplasm. In case you don't know before, please note this, they can ask you. Protoplasm is cytoplasm plus nucleus. Is that taken? Then your cytoplasm is just like saying cytosol plus organelles. So the cytosol and all of the organelles, so the cytosol is the liquid. Then all the organelles that are present in the cytosol forms your cytoplasm. Quickly. Between the contractile vacuole, you know you have the contractile vacuole at the one at one end and the other one at the other end. It is the food vacuole that will take it to the pore, so the waste will be released out of paramecium. So it is the food vacuole. Is that taken? The contractile vacuole just take it, then expel it. But the food vacuole take it, then there's an anal pore. There's what we call anal pore, where the waste are released. Can we move on? The rod of the retina is what? Is a cell, Abi. So is there any question here that is confusing? Glancing through it. Oh yeah, quickly move to the environment. Number 1. Either way, you will see one question or two out of this aspect. And if you don't see, good luck. We'll continue with where you will see soon. So this can just be like your revision notes. Oh yeah, number 1. A spirogyral cell was placed in solution X. After 1 minute, the cell has plasmolysed. Solution X is what? It should be a very strong sugar solution, Abi. Why? Because the sugar concentration is more outside. That's why it is taking water from the spirogyra, and the spirogyra plasmolysed. Do we all understand that? Number 2. Movements of molecule from higher concentration to low is what? Diffusion. Number 3. A fresh kola nut, 1 gram, was out into salt solution. After 3 hours, the kola nut weighed 1.2 grams. The simple experiment demonstrated that the salt solution is what? So it has gained more water, B. So that salt solution must be what? Hypotonic to the cell, Abi. Do we agree to that? It went from 1 gram to 1.2 grams, so it has gained more water. So that means that the solution must have been hypotonic, so that it now takes water from the solution. Are you there? Number 4. The filament of spirogyra were put into equal volumes of equal. I think since you can answer 1, 2, and you should be able to answer number 4 if you calm down. So anyone, let's go on. You should also be able to answer number 5. Number 6. Let's answer number 6. If an isolated living cell is left in distilled water for 2 hours, it is likely to what? Isolated living cell. What will happen to it? Eh? That's why I said we should do it. It will become turgid. Forget about death or no death. We are not specific here. It will become turgid. Is that taken? Because it will take in water and become turgid. Let's be careful. Biology questions can be trickful. Is that taken? Please, not die from excess water. Some cells might even find a way to regulate the water, and they are not specific about which cell it is. So the only thing we know is that it will take in water and become what? Turgid. Is that taken? So what's the answer to number 7? Polythene, Abi. Number 10, what's the answer? Mineral salts can be absorbed into the roots by what? You can answer number 9, Abi. Now, mineral salts are present in the soil. So they move through from the roots to the body of the plant, up the stem. Water from the soil moves in by osmosis. But those ions move in by diffusion and active transport. Active transport, I already told you. The ions that are moving in from the soil, they are more than the ions that are present in the roots. But still, the roots need those ions. So the mechanism is that those ions will now move from the roots, from the soil into the roots. And when you move from where it is more to where it is less, it is what? Active transport. What's it called? That's from where it is less to where it is more. Sorry, that's what I wanted to say. So in the roots, you have more ions than in the soil. Yes. So it moves from the soil down into the roots. Then that is why you have what we call active transport. But as those ions are moving, carried by, there are different parts of the membrane. There are channels. There are what we call the ion channels. There is potassium channel, there is sodium channel. They will just move within those channels. So they diffuse through those channels. Is that taken? All these channels, well, you still talk about them later, but for your level, if there is time, you are supposed to actually learn it too, because that's how you understand some part of biology too. There's what we call potassium channel, and there's what we call sodium channel, and there's what we call sodium potassium channel. All of these things are, it's in your modern biology too, so don't look at me like. So you should be able to answer number 11 if you calm your body down. Number 12. What's the answer to number 13? Hemolysis. I think you should be able to answer the rest. What's the answer to 19 and 20? Please, if they ask you in the exam that shrinkage of red blood cell is what? It is crenation. Don't make mistake and pick plasmolysis. Adeshewa, ọmọ Yaruka ìtútù bẹ̀rẹ̀ sí ń sùn. Òtútù ń sùn. Who is that? Wọ́n máa bá yín sọ̀rọ̀ kẹ́ ẹ̀ prepare ara yín fún class. So please, should we just move on from classification to nutrition? Yes. So for nutrition, is there anything you don't even know in nutrition? Maybe for revision sake. Ẹ bá wá aṣọ gidi torí ọlọ ọwọ́ ń ro mí fa. So please. So hello, every one of you know what we call nutrition. Process by which organisms obtain, search for, obtain food, take in the food, digest it, and assimilate the food for use. When you are talking about nutrition, we have what we call autotrophic nutrition, Abi, and we have what we call heterotrophic nutrition. Now, for autotrophic nutrition, you already know they produce their own food, Abi. An example are your green plants. Now, autotrophic nutrition can involve photosynthesis and can involve chemosynthesis, Abi. What's the difference between the two? Photo means lights. When the manufacturer, the autotrophs, they use lights, they use CO2, they use water to produce their food, and that food is glucose, Abi, which will be converted to starch, Abi. Then when they produce glucose, they produce a byproduct, that is oxygen. So this process is called photosynthesis. In general equation, CO2 plus H2O in the presence of sunlight to give glucose plus O2. So the process in which living organisms manufacture their own food in the presence of sunlight using CO2, water to produce glucose and oxygen as byproducts. So in case they ask you that what is the byproduct of photosynthesis, it is oxygen. So an oxygen as byproduct, then what we call it photosynthesis. Now, photosynthesis takes place in two stages. The first stage is called the light stage. Are you there? The second stage is called what? The dark stage. Now, for the light stage, you need to know where it takes place. Light takes, please, dark stage just means that you don't need UV lights. It does not mean that it occurs in the dark. It can occur during the day, but you don't really need the UV lights. That's why. So dark stage, now light stage, light stage occurs in the chloroplast. That place where it occurs is called the grana. I hope we know that chlorophyll is needed for this process. These are the things that you all should have known by now, but in case you don't know. Light takes place where? In grana. Grana is called stack of thylakoids. Stack of thylakoids. That's grana. Then the dark stage takes place in what we call stroma. So you might see grana in your option, you might see thylakoid in your option. Know what to go for. How you remember this is LG. Just remember LG TV. Then that tells you where it takes place in light stage takes place. Now, what are the things that happen during light stage? Quickly. Number one thing that will take place during light stage is absorption of UV, absorption of sunlight by chlorophyll. That's what will take place during the light stage. That's the first thing. Absorption of sunlight by chlorophyll. Then that sunlight that is absorbed will now split water. So the second stage is called photolysis of water. And during photolysis of water, let me put it this way, A2O to give H plus OH minus. So the light will help us to split water. So we call it photolysis of water. Then all of these guys will come together. Then at the end of the day, oxygen will be produced. So if they ask you that the oxygen formed during photosynthesis, where did it come from? It came from water. Is that taken? Let me just do it this way. OH minus plus OH minus, so to say. There are a lot of them. Then you have A2O plus O2. But that's just by the way, just for you to remember. So when water photolysis or photolyze, so to say, you can put it in your dictionary. It gives oxygen. So during photolight stage, oxygen is produced. Another thing that is produced alongside is ATP. Adenosine triphosphate is also produced during light stage. Another thing that is produced is the NADP. NADP plus. It will take hydrogen that is produced here and it will form NADPH. So all you just need to know is that number one, where does light takes place? Ghana, Abi. Number two, where is oxygen produced during the light stage? From water, Abi. Now, another thing is that what is the first thing that will take place during light stage? Then last thing, what are the products of light stage? ATP and NADPH. Is that taken? Oxygen is a byproduct. Do you get that? Now, let's move on to the dark stage quickly. Dark stage takes place where? In the stroma. For you? For you? I'll buy you another one. I did not promise you. Have you written? So now, dark stage. So the thing you need to know about dark stage is that number one, it takes place in stroma. Number two, the products of light stage will be used. So that ATP you produce and that NADPH will be used in the light stage. So these ones are used in the dark stage. Dark stage is also called what? Calvin cycle or light independent stage. Light independent stage. So now, what are you now going to do? CO2 that you absorb. You know in the reaction we said we have A2O plus CO2 in the presence of light to give glucose plus oxygen, Abi. And we have used water already. We have used UV light. So what are we going to use? CO2 during that stage. That CO2 reacts with the hydrogen carried by this guy there, carried by NADH. So CO2 plus the hydrogen eventually will now give us glucose. So what takes place in that stage is the reduction of carbon dioxide using hydrogen from where? NADPH. Is that taken? That's what takes place during that stage to produce glucose. And you have produced the oxygen before. So that's the process of photosynthesis. Am I good to go? So, can I rub the board now? So for chemosynthesis is the manufacture of food using inorganic substances. So some organisms, they use inorganic substance to get their energy, and that is called chemosynthesis. From the name chemicals. But don't make mistake when they ask you, it is not organic chemicals, but inorganic substances. So chemosynthesis is the oxidation of inorganic substances. Is that taken? That's chemosynthesis. Can I rub the board now? Everywhere. Somewhere, nowhere. Thank you. So now for heterotrophs, you know what heterotrophs do, right? So they depend on autotrophs for their food. We'll solve all our questions together. So they depend on heterotroph for their foods. And most times, the one that feed on solid materials are called the holozoic, Abi. But we have what we call omnivore. We have the herbivores, Abi. And we have the carnivores. Now, quickly, you know the difference, right? This one feeds on herbs, on plants only, Abi. So they feed on plants only. Omnivore feeds on both what? Plants and flesh, Abi, and animals. So that's what they feed on, omnivore. Why carnivore feed on flesh, Abi? So they feed on flesh. Everyone knows this. Now, if you're not talking about, you know your herbivores, you can give examples. You know your omnivores, you can give examples. But your carnivores, we have carnivorous plants. And we have carnivorous animals. Abi, there are a lot of carnivorous animals. What are the common carnivorous plants? They can ask you. Sundew, pitcher plants, liverworts, wow. Bladderworts, Venus flytrap. Please, I don't know who has college biology here. I think there's a diagram of these plants and it's labeled Idodoume. Please, before you go for your exam, especially those of you that want 90 something, study that structure. No study, just look at it. I don't know if you have Idodoume. Let me show you something. Hurrah. Oh yeah, Idodoume. Let me quickly show you something. There was a question they asked in JAM last year, and they can ask you this year. You will see the way the plant looks. Oh yeah. Can we move on then? So you go and check it yourself. When you see Idodoume, check all these plants. Check their labeling. The parts that are leaves, flowers, and all of those things. I've forgotten the one that was asked. That's why I want to quickly check for you. But you can check yourself. Now, all these plants, the major reason you feed, they need, please, they need nitrogen. But these organisms cannot really get nitrogen. So in order to get nitrogen, they have to kill animals. So they feed plants that feed on animals. So they will set their trap and they will trap the animal, then they kill the animal, then they absorb nitrogen from the animals. Then that is why they are carnivorous. Is that taken? You know your animals that are carnivorous. Now, when you're now talking about feeding relationships, show me one class. Can I rub these parts? So now, all these organisms, they depend on one another, microbia and all of those things. So you have what we call feeding relationships. And all of you know this. Feeding relationship, when organisms depend on one another, either negatively or positively, we call it symbiosis. A lot of us make mistakes. Symbiosis is different from mutualistic relationship. Mutualistic is when both of them are benefiting. So all those relationships you are talking about, they are called symbiotic relationships. So your symbiotic relationship can now be commensalism, parasitic, mutualistic. Do you get the point? But in case you are asked very good. This was the one they asked in JAM. They just drew it like this. Then they now put all this label. They now said this one, what is it? Is it leaf or flower? Do you get that? It's in your college biology. When I saw it in my own exam. And normally before I really did not study all this thing. So when I got to exam, I saw it. I was now wondering that they wrote in the options flower, leaf, all the parts of plants. I was now looking down, does this guy look like plants? So I had to be using brain that if this is the stem, I think this can be the flower based on the way it looks. You can see flowers should not be more than leaves. So if you have sense, and I said it must be leaf, and I chose leaf. When I got home and I was looking at all of my textbooks, then I saw the diagram and I said leaf. Nice one. So you don't leave your brain at home and go to exam hall. It's not everything you might have seen before. That's why you read different textbooks when you are preparing. As you are reading modern biology, you finish reading it, all the important points there, you go to essential biology, same topic. All the important points there, you go to college biology. All the important points there, you go to your normal. This is Idodoume, there's still another college biology. So gbogbo nǹkan tó wà níbí báyìí, and you can study the rest of them too. These ones are not labeled. You can study these ones. Just study the labeling before you go. All those are strange questions that anybody can see and feel like they might not really know. So just note that. Now, for feeding relationship, we call it symbiotic association. Symbiotic association. Are you there? Yeah. Then we have what we call a parasitic relationship. We use positive when an organism is gaining. We use negative when an organism is losing. And we use zero when an organism is neither gaining nor losing, neutral. Is that taken? So now, for parasitic, there's one organism that is gaining and there's one organism that is losing. So the type of association is plus and minus. Is that taken? It's living there, it's feeding there, and it's destroying this guy. So that is what we call parasitic relationship. Now, there are two types of parasites. We have what we call endoparasites. Let me say parasites, animal parasites, and we also have plant parasites. Are you listening? For the animal parasites, we have what we call endoparasites. They are parasites that are inside, find within, not on top of. So, endoparasites, examples are your tapeworm, Abby, all those worms. They are your endoparasites. They are usually found within. Tapeworm, guinea worm, roundworm, all those ones. Now, we have ectoparasites. Ectoparasites are found outside, Abby. Yes. And that is your tick, your lice, and so on. So all those insects that, bedbug, all those insects that you have on top of your body, in your ear, that they feed on your body and you are negatively affected, then they are called ectoparasites. So then we have some plants too that are parasitic plants. These plants feed on another plants. Is that taken? Example of parasitic plants are what? Let me slide through. You can spell it well. So these are parasitic plants. And what makes them parasitic is that Please don't forget this. What makes a plant parasitic is due to the presence of Ostoria. Ostoria. Are you there? So can we move on now? So parasitic association is a plus or minus association. Next one, mutualistic. Another association is mutualistic. And from the word mutualistic, you already know what that means, right? That means both of them will gain, Abi. So it's a positive and positive association. You give, I give, then we gain together. That is that. Now, most importantly, you need to know the examples associated. They can ask you that the association of fungi with the roots of leguminous plants is a type of which association. That's why you need to know all of those examples. So one of the example of mutualistic relationship is mycorrhiza. That is an association of fungi and roots of higher plants. Roots of higher plants. Then we call the association mycorrhiza. It is mutualistic. The fungi cannot produce its own food. Plants produce food. But the fungi helps the plant to absorb nutrients from the soil and give it to the plants. Are you paying attention? Yes. Because they will ask you that what is the function of the fungi in the association? What is the function of the plant in the association? The plant supply food to fungi. Food, carbon, and all the necessary things needed by fungi. Why fungi helps it to absorb nutrients, supply nutrients. Ele might take nutrients from soil and from plants so that plants will use it to produce food. Is that clear? Now, there's another one. We have what we call the rhizobium. Rhizobium at root nodules of leguminous plants. Root nodules of leguminous plants. If you remember another example, you can let me know. Now, you remember what rhizobium used to do at the root nodules of leguminous plants? They used to absorb and they used to fix nitrogen, Abi. They are nitrogen fixer. We will get to nitrogen cycle as we are moving on. So they are nitrogen fixer. They fix nitrogen in the plant. Then what will happen to the plant too? They supply nutrients to and they supply house too. The nodules of those plants is where these guys live, and they continue to supply food to these guys. Which other example? Hermit crab and what's that one called again? Sea anemone. Is it mutualistic? There is what we call hermit crab versus sea anemone. All these examples are important. That's why I'm trying to help you to remember them. Hermit crab and sea anemone, they go together. Now, the sea anemone has like tentacles, stinging cells that it will be on top of the crab. The crab will be the one carrying it everywhere. Is that taken? So then it will be helping the crab to carry out defense, majorly defense, so that nothing attacks the crab. Then the crab also carry the anemone everywhere in order to get food. So that is a mutualistic association. During post-UTME, especially for those of you going to OAU, there are still some associations under here that they have a specific name. Although last year, OAU did not ask those questions they used to ask. They asked normal questions last year that are still somewhat technical, but for a brainy, you should still be able to answer them. They are not tough. But when we get there, we'll cross it. Now, the next thing is, which other example can you remember? Flowers and pollinators, Abi. Which other example? Oya. Remoral fish is commensal. Which other one? Yes. Lichen. Leaching. That's fungi and what? Fungi and algae. So let's move on. We have a long way to go. So now, the next one is commensalism. When do you have commensalism? Oh yeah. I'm doing digestive system now. Commensalism. In commensalism, one gains, Abi, one is neither gain nor losing. Is that taken? An example is your remora and shark, Abi. Shark and remora is an example. The shark continues to feed, and all the crumbs from the food the shark is feeding is left for the remora fish. And this association at a stage is said to be mutualistic due to one or two things. But for now, let's forget the one or two things. Now, what other thing? What other example? Epiphytes. Epiphytes is a plant that grows on another plant. This plant lacks what we call ostoria. Lacks ostoria. It grows on another plant to survive. So it lacks ostoria. If it has ostoria, it would have been parasitic and not mutualistic. This plant only needs sunlight, so it needs the plant to go to where it will get sunlight. Then that is why it is called an epiphyte. But if it had ostoria, it would have sucked. It would have sucked out all the nutrients from the plant, and the association becomes parasitic, but it lacks ostoria. So now, which other example? Sick bed, Inko. Cattle. One of them is mutualistic. Cattle and sick bed, Abi, is commensal, Abi. There's one other one. This one is mutualistic, Abi. There's one other one. Yes, that's oxpecker. Buffalo. Buffalo and oxpecker. This one is mutualistic. Yes, this one is commensal. This one is mutualistic. So there's one that used to help this cattle to get sick on top of the cattle. So it gets sick. All the sick ectoparasites on the cattle that is feeding on the cattle's blood. So the bed will be picking all those things. But in this one, the cattle, or what is it called? As it's feeding on grass, it's uprooting the roots of these plants as it's feeding on the grass. So all the insects that are present in the roots, I don't know if it's Pekka or X-ray, but just check it. This bird will be feeding on all those insects. And as it's feeding on all those insects, it has no effect on the cattle. Is that taken? The cattle is feeding on the grass. While it is uprooting the grass, some insects are being uprooted. The bird will not be feeding on those insects. So it's only the bird that is gaining. It has nothing to do with the cattle. Do you get that? That's why that is commensal. But the one of the bird, I think is oxpecker, that is feeding on the back of the plant and the animal and feeding on the tick, is getting his food, while it's also helping the animal is also getting rid of parasites. Then they are mutualistic, living. So compare these two and place one as the first example I said and place the other as the second example I said. So can we move on? So which other example? So we have what we call amensalism. Amensalism. One is losing and one is not affected. That is amensalism. So amensalism is almost like opposite of commensalism. Is that okay? So we have amensalism. Amensalism. In this case now, they asked this one in JAMB last year, but the way they asked, they used I think antibiotics or something. So listen, in amensalism, one is not losing and is not gaining, and one is losing. Nothing is positive here in the association. A microorganism release antibiotics or anything that is toxic in its environment as a waste product. Then other organisms growing there, they continue to die due to the presence of these antibiotics. It is one organism that releases it as a waste product. Then one, because of this thing, this one is also present there. But because of the presence of antibiotics where this organism is living, then this organism dies. So at the end of the day, the one that releases waste is not dying, but the one that is present is the one that is dying. Is not gaining from the waste, but is getting injured from the waste, and that is what we call amensalism. So there are some things that some organisms can release as their own waste products, and it will have negative effects on another organism. Then we call that amensalism. Is that okay? Eh? What we are saying is that there is penicillin, Penicillium notatum. It's an organism that releases penicillin. Abi? Now, when it releases penicillin, penicillin is used to treat bacterial infection. Now, the penicillin produced is not using it. The organism just produced it. Let me put it this way. Let's say you pollute, and your own pollution is not affecting you, but it's affecting your neighbor. But that's not really the sense, because it will affect you too. But if one organism is secreting substances that has no effect on that organism, but that substance has a negative effect on another organism in that same environment, then that is called what? Amensalism. Do you get that? Substance released by one organism is not kiniko. The egrets are the leke. Yes, they are the leke. Is it true that they eat sticks on cattle? Yes. Egrets are the ones that used to be on the ground though. So the commensalism should be egrets and cattle. The mutualistic should be oxpecker and this buffalo. Yes. Egrets and cattle. Egrets are on the floor. All those white birds. So as you are uprooting, please check that and confirm that later. Is that taken? So let's move on. So we have talked about parasitic association, mutualistic association, which other one? Commensalism, and that's what I just said now. Cattle and egrets, what did I say they are? Commensals, Abi, and that's what's. So why buffalo and oxpecker? They are what? Mutualistic. Thank you. Is that confirmed now? And you know the difference. One is getting thick, while the one is staying off disease. So, but this one, as the cattle is uprooting, all those white birds are feeding on the insects, and that is just commensalism. Are you there? Can we move on? So let's look at the digestive system quickly. I'll summarize the digestive system now. So that we'll do all the questions on nutrition together. Standard living. So for digestive system, holophytes. What's the difference? Did I not talk about holozoic? And it's still, I think holophytic is focusing more on plants. Holophytic and holozoic. Holozoic is the solid part. And I think they are, I don't want to say they are the same. But let me check. Let me check holophytic and holozoic again. Please, let's move on quickly. Now, when you carry out nutrition, you are no longer interested in the class. Maybe you know everything already, so I should be going. Please take note of these things you are doing. It should be in the back. It should be in my back. Then if be watching if battery. So please, can we move on? You're already stressing me the way you are talking. Why are you people moving up and down? We still have a long way to go. Yes. So thank you. It can also be amensas. They secrete those chemicals to get rid of substances around them, organisms around them. Oh yeah, have you checked what I said you should check? Holophytic. So holophytic is still like autotrophic nutrition. Holozoic is heterotrophic. So please, let's quickly move on to digestion before we solve questions. Now, for digestion, when you carry out nutrition, there are some steps you go through for nutrition. One is you feed, ingestion. I don't know if this spelling is correct. You ingest food, you take it into your mouth, then you break down the food, then we call that digestion. Then when you digest the food to the absorbable forms, the ones your body can use, we call it absorption. Then when your body starts using the food to produce all the necessary things your body needs, we call it assimilation. Then at the end of the day, the ones that your body did not use, then we call it, is this correct? So please, nutrition involves these steps: ingestion, taking in food; digestion, breaking down food; absorption, absorbing food; assimilation, making use of absorbed products to get what you want; then ingestion, releasing the things you don't want. So now we are focusing on the digestion. So whenever you take in food now, we have talked about how you can get food, either through mutualistic behavior and all of those things. There's nothing to be funny about now. we might have been joking with some things, it's biology. But for now, leju pani. So there's nothing funny in this class now. Learn what you will learn, solve the question you will solve, and move on. That is what we are doing. So for ingestion, you've taken in your food already. Then you need to break down the food. So the enzymatic degradation of food. So the enzymatic degradation of food is what we call digestion. Now, where does digestion start? It starts from the mouth. So at the mouth, first thing that will take place is what we call mastication. Mastication. All your teeth and mouth, they start moving, they start crunching the food. So I don't know what simple English to use for mastication. Chewing. Thank you. The white guy. So you start chewing the food and you start mixing saliva with it. There are different saliva you have. The glands that produce saliva, there's one at the upper part of your mouth. That one is called the parotid gland. Then we have one under your tongue. Then that one is called sublingual gland. Then you have one at your jaw, around your jaw area. We call it submandibular gland. Now, all of these glands, what do? They give saliva, Abi. Now, saliva contains an enzyme. The saliva contains an enzyme. And that enzyme is, do we all know saprophytism? Yes. So I've talked about the rest of the association, but we forgot saprophytism. So saprophytes are organisms that live on dead, decaying organic matter. So saprophytes feed on dead, decaying organic matter. Is that taken? Anything that has died, not like vulture now, they feed on it. They start breaking it down. So saprophytes usually carry out what we call extracellular digestion. You need to note that. And examples of saprophytes are fungi, bacteria, which other one? Nematodes. Is that taken? So they feed on this hydra, coelentrates. Are they saprophytes too? All these mushrooms are saprophytes. So please, is that taken? Are we following the class? Or am I just talking to myself? Because you are making me to scream. So at the mouth now, the glands produce saliva. You chew, Abi. Now, when you are chewing, you use some things to chew. Then we call it teeth. Your set of teeth is your dentition. So your dentition. Now you have milk teeth at a certain age in life. Those milk teeth or temporary teeth, or what else do you call them? Those, we also call it deciduous. Torí wọ́n máa wọ́ dànù. They will ré dànù. Deciduous teeth. That set of teeth, they are 20 in number. Then after a while, you start bringing out new sets. Then we have what we call permanent teeth. Abi? Permanent. Permanent are many. 32 for humans. For us, there are 32. The 32. For dentition, we have what we call dental formula. Dental formula. Abi? For man, our dental formula is incisors, canine, premolar, and molar. So you're going to have upper incisors, lower incisors, upper canine, lower canine, upper premolar, lower premolar, and continue. Abejoro. So for the incisors, canine, premolar, and molar, you now have this one to be 21, 23. So just remember 21, 23, that is human. So that will now be 21, 21, 23. That of dog is what? 31, 43. You can even open it for me. So that is the dental formula. Now if you look at this, 2 plus 1. Put you need 2. 3 plus 5 is what? 8 plus 8 down. So that is just on one side. One side, if you divide your bilaterally, so on this side, you have 16. 8 up, 8 down. On this side, 8 up, 8 down. So when you count this, you multiply by 2. That's what gives you your dentition. Is that taken? I know some of it is not complete again. What's buying your? So now, we can also talk about the structure of the tooth. I really cannot remember the structure again. So please, we need to know the structure of the tooth. I really cannot remember vividly like that. I know it has roots. I know it has the layer. Then I know it has all those vessels, Abi. So please, help me with this. The structure of the tooth and the labeling. The upper part is the enamel, Abi. So you have the enamel, you have the dental, and so on. You have the gum and all. So please help me with that structure. They can ask you. So now, all these structures, what are the functions of the tooth? And again, we have 2 types of tooth. Depending on the sets. Enamel, dental, gum, neck. Go and draw it, is in your book. Olele. The first part is the enamel. It's very hard. Dental. The dental is supplied with blood vessels. Then you have the pulp cavity. Pulp cavities are the blood vessels. Then you have the gum. And it is always painful when you have injuries on all these parts. So look for the dentition of man, rabbit, and dog. Just remember 21, 23. For dog is 30, 31, 42, 31, 43. That's for dog. For rabbits, 20, 33. Have you written it? For dog, it is what? 31, 42, 31, 43. Why for rabbit? 20, 33, 10, 23. And remember, that 0 part is the what? Canine. Because herbivores feed on grasses, so they don't have canine. They just need to cut with their front teeth, and they will start crushing with their jaws, with their molars and premolars. And that is what they do. So, and they are called carnation. Carnation is the dog, the one that has the long canine. So please, based on the sets, the type of set of teeth, don't suppose you tell me, oh, you will start stressing you. You will start knowing acrodonts, theodonts, and all of those things. But for now, based on the same, if you have same set of teeth, and that is seen in all these reptiles, please, you are talking too much. That question was asked last year, that what is the set of teeth seen in reptiles, in lizard? It is homodonts. So when you have same set of teeth, it is called homodonts. When you have different sets of teeth. It is called heterodont. So quickly, what is the function of incisors? What is the function of incisors? So you cut or bite with incisors. What's the function of canine? You tear. That's how you used to eat your meat. Premolar. Almost the same thing as molar. Molar is what? So you crush it like you crush the bones. So they can ask you for the functions of this guy. And don't forget, they ask you how many incisors. Just know that these are incisors. How many in total will it be? You have 2 up, 2 down. It will be 8 in total. Is that taken? If you multiply it times 2. So now let's move on now with the digestive system. Characteristics. Is that not what we just talked about now? Which? Okay. This one is, yes, incisors is flat. I think you asked in mock. Yes, I remember some of it. Flat and chisel. This one is long, pointed. This one is pointed and sharp. You know how it looks now. What would be the description for this now? Rigged. Flat and rigid and large. So please just note that, but it's something you should be able to think about. I'm sorry. See you. Keep. Now say cups. Cups. I'll read this one silent. Cups. Oh yeah. Please. Are we okay with dentition now? Please, these are the questions they can ask you on dentition. Saprophytic, is it related to decomposing? Yes, it's related to decomposing, but it's not necessarily that saprophytes are decomposers. Most saprophytes are decomposers, but not all decomposers are saprophytes. Which one is symbiosis? I already said the association in general is symbiosis. What is the substrate taken in? Saprophytes, they take in all the possible nutrients. They take in starch, they take in glucose. If it is fungi, then it will be stored as glycogen. Is that taken? Fungi store food as glycogen. Between molar and premolar, which one is missing in the deciduous teeth? It is the molar. You don't have the molar in the deciduous. Is it molar or premolar? So please, you don't have molar in the deciduous teeth. Shegbo, what's the difference? Wait. It is molar. 3 plus 3 is 6. 6 times 2, 12. 32 minus 12, 20. So it is molar. Oh yeah, duster. I want to continue with the degradation now. The food has left the mouth now. Where is it going to? The stomach. Can I rub this? We will finish up biology today or else we will sleep here. Everything biology. Because me, I cannot sacrifice my life and come here and you will still not do anything. We will still carry the textbook and I'll show you some parts. Let there be time. And don't make noise, please. Oh. So the food has left the mouth now. The food is going to the stomach. But before it gets to the stomach, it will pass through the esophagus. There is no digestion in esophagus, so shebi you know. So there's no time, but after the stomach, then you move to the, I'll just put the stages from the mouth to the esophagus. Please, you are talking too much. Problems in here. What takes place during the esophagus is what we call the peristalsis movements. Peristalsis movements. Now there's an opening. Peristalsis movements through the esophagus. So you have swallowed the food now. So the food is moving across the esophagus. Now, it's premolar. That's why it's because of the calculation that I just used to conclude. Please. There are some animals that you will see that there will be a space in between. There will be one teeth here, there will be space here. Please confirm that later. Confirm that later. Is that taken? Please. Textbook says molar. AI says confirm it later. Can we move on, please? We still have a long way to go, and I'm the one shouting. Are you done? Can we move on? Some of you will not listen. So the movement, the muscular movements of the food down the esophagus is called peristalsis movement. Then when that food, it will pass through a muscle around here. That muscle or an opening through which the food will go into the stomach, we call it pyloric sphincter. That's what we call it. Don't worry. I didn't have much time to prepare for the class. I would have prepared better for you. There's one that comes. The first one should be cardiac. It's one that is close to the heart. Yes. Why the second one is? Are you sure? Oh yeah, let's move on. So we have this cardiac sphincter, and when it moves through the cardiac sphincter, I remember the one that is up is the cardiac, but now when it enters the stomach, then the food will stay there for a while. Then what will happen to the food in the stomach? Number one, at the stomach, HCl will be produced by some cells. Those cells are called parietal cells. So they produce HCl in the stomach. HCl is in the stomach. So what is the function of the HCl? Number one is that it creates acidic environment. But the major function is that it helps to get rid of pathogens. There are some microorganisms that you might have eaten with your food. So the acid will help you to kill them before they go on with to destroy lives. So another thing that is present in the stomach is pepsin and renin. Is it the single L or the double L? Single L. So there's pepsin and renin. So what was broken down here? I think we didn't finish this. I said in the saliva you have which enzyme? Ptyalin. Ptyalin will help you to break down starch. So it will help you to break down starch. It's from starch to what? Maltose. Is that taken? So ptyalin helps you to break down starch to maltose. Then, when only starch is broken down in the mouth, in case they ask you. Now, when you get to the stomach, you break down protein. So digestion of protein starts in the stomach. Is that taken? Now, pepsin will help you to break down protein to peptones. So peptones, are you there? While renin helps in what we call the curdling of milk. It helps you to curdle milk. And curdling of milk takes place by converting I don't know if the spelling is correct. Carcinogen to casein. Please, you know what I'm saying? It converts carcinogen to casein. Spelling is a great error in biology, but for now it's pardon. We want to go the old syllabus. So for now, I know carcinogen. This is insoluble. This is soluble. Is that taken? So it converts the soluble carcinogen to insoluble casein. So that is what happened in the stomach. Can we move on? Now, when you leave stomach, now where are we going? Small intestine. All those you used to eat. So now, when the food is leaving the stomach, what do we call it? Chyme. So the bolus that is leaving the stomach, so we call it chyme. Are you following what you are doing? Yes. So now the food that is about to leave the stomach now in the meshy liquid form, that is called chyme. Then when it gets to the stomach, there is the liver. Are you there? Then around the stomach, there is the pancreas. Is anybody calling me? Then with this liver, there is a structure called the bile ducts. Are you there? I mean gallbladder. So this gallbladder, the liver will produce something called bile. The bile will be stored in the gallbladder. There is no space to draw, but can you see what I'm doing? Yes. This thing that is attached to the liver is called the gallbladder. That is where the bile will be stored. Then when the food is coming, there will be a common opening from the gallbladder to a region around the small intestine. Then the pancreas too. So these ducts that comes from pancreas, we call it pancreatic ducts. Then the one that comes from the gallbladder, we call it the bile ducts. Is that taken? Then at this junction now, pancreatic ducts Pancreas will release what we call pancreatic juice. So let me go on to split it into three. So when it leaves the stomach, it gets to where now? The small intestine. Listen, oh. The small intestine is three. Duodenum, jejunum, and so duodenum, jejunum, and ileum. Now let's go on, please. Now duodenum is the first place. That's this region. Now, what happens to the duodenum? We come from gallbladder, Abi. What's in the gallbladder? Bile. Then the bile goes through the bile ducts, Abi, and it is deposited wet in the duodenum. Abi, are you there?
Yes.
Then another thing, we have the pancreas. What comes from the pancreas? Pancreatic juice. Then pancreatic juice will go through the pancreatic ducts. That's why this kind of gland now, they are called duct gland. They ask in exam too. There is difference between duct gland and ductless gland. Anyone that has passage, that you go from that place to where you are going, then we call it ducts. When we get to hormones, I will remind you again. But for now, understand that there is difference between ducts and ductless. Are we there? So now, the pancreatic juice, there are different pancreatic juice. Where is it also going? Duodenum. So what is happening at the duodenum? Don't forget the cardiac sphincter that opens to the stomach. So the pyloric sphincter will now open from stomach to the intestine. So now, at the duodenum, what will the bile do? Bile will cause emulsification of fat. You know, since morning now, we have not broken down fat at all. So the bile will first help you to, it's not breaking down fat, so it's helping you to just break it into globules. It's still fat. It's like when you put soap in fat, or you put something and fat breaks to small portion, but it's still fat. Okay, something like that. So now that bile, aside the emulsification, it also provides alkaline environments. You remember that where it is coming from is acidic, Abi. Then when it gets to the intestine, it needs an alkaline environment. That's where other enzymes will work. So what are the enzymes? Those are the pancreatic juice. So the pancreatic juice, you know, the pancreas can still produce hormones, but we'll talk about that later. So the pancreatic juice, those are now your amylopectin. This one will break down your maltose, your what's it called? Your sugar, starch to maltose. Is that taken? You also have trypsin. Trypsin will break down your peptone to polypeptide. Are you there? Are you following?
Yes.
Then, which other one? Lipase. Lipase will break down your fats and oil that has been emulsified now. It will break down fats and oil. It will break it down to glycerol and fatty acid. So if they ask you, the absorbable form of of starch is glucose. The absorbable form of fats is fatty acid and glycerol. The absorbable form of protein is amino acid. Are you there? Those are the ones you will make use of. So you are still breaking them down. But you would have had some amino acid now. Then when your thing will now leave, your food now leaves duodenum, it now moves to jejunum. At the jejunum, some part of the food will be absorbed. Then other this thing will be secreted, other enzymes that you need to now completely break down this guy. So can we go on now? Can I rub this part? Please write it in a way that you will understand. So what happened at the jejunum? So at the jejunum, there will be absorption, not really absorption per se. The last this thing takes place, nothing much takes place at the jejunum like that, but some part of the absorption will take place. Then I've forgotten. There's one of these guys that will be broken down. But let's go to ileum first. Ileum is where final digestion and absorption takes place. So at the ileum, you will have all those your normal enzymes. Maltase. What will maltase do? So maltase will help you to break down maltose to glucose. Then glucose will now be absorbed. Which other one again? We have the repsin. Abi, the repsin will break down polypeptides to amino acids. Are you there? Then which other one? Still your lipase will break down the remaining fats to fatty acid and glycerol. Now, all of this food now that you have broken down will be absorbed by your body. Then what absorbs it? There's a region of the ileum. That region of ileum where food is absorbed is called villi. So food absorption takes place at villi. Is that taken? So I think to some extent. Then when you now leave the small, everything we have been saying since morning is happening where? In the small intestine. Abi, we can confirm this one. Nothing much takes place here, but there's one thing that skipped my mind. So I'll confirm that. When you now move from small intestine to large intestine, then that is where you have water reabsorption. Is that taken? So all the water, all the things that are left in your body now that you did not break down, all the part of the bag, bilirubin, mixed with your shit all together. Then your feces, you now pass your feces out of your body. Then that feces you pass out will go out through your anus. But at large intestine, they ask you what takes place. At large intestine, absorption of water. Is that taken? So the remaining food will not be had. Our amarojun toilet. So all the water from those part of the So can we move on? Do we understand where we are? So now, then you have absorbed the food now. Then you assimilate. All those things, glucose will now be taken to mitochondria, where you are going to use it to produce energy. Shey you get? All those process now is now what we call assimilation, making use of all the things you have absorbed. But we will talk about that next after transport system. I'll quickly talk about transport system, all the things that are needed for transport. So can I rub the board now? So moving on from here, we have, when you now release all the waste products, we call it what? Ingestion. So what are the classes of food we have? That's the next thing. Those are the areas they can ask you questions. We are still on nutrition, no? Then you talk about nutrients. The most important elements for life. Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. We call them schnapps. And these elements are found in different molecules. But the biomolecules we have for life, they are, let me list them here. Carbohydrates. The formula for carbohydrates is CX A2Y A2OY. So that's the general formula for carbohydrates. We also have proteins. Then we have vitamins. We have minerals. We have water. And we have roughage. Now, for carbohydrates, your carbohydrates are generally made up of carbon, hydrogen, and oxygen. They are the main source of energy. Main source of energy. That's where you get energy the most. And they are classified based on the number of sugars they have. We have what we call monosaccharides, Abby. We have what we call disaccharides, and we have what we call polysaccharides. Now, monosaccharides are usually your glucose, galactose, which other one? Fructose, mannose. So these are your monosaccharides. They are simple sugars made up of only one unit. Disaccharide too. So they can ask you in exam, maltose. What will maltose give you? Disaccharide is made up of two monosaccharides. So maltose will give you what? Glucose plus glucose. Is that taken? Which one is next? Lactose. Now they can ask you that where do you find this guy? From the word maltose, maltina, malt, lactose, milk sugar. So lactose will give you galactose plus glucose. Then you have sugar cane. We call it cane sugar, coming from sugar cane. This is made up of glucose and what? So please, one of the questions that is so common is the test for this food. How do you test for carbohydrates? We use test, Abi. Failing A and B. We can use Stone's reagents. Which other tests? We can use Benedict's solution. I think Benedict's solution is also Stone's. So please, let's know about these tests, especially the color changes. The color changes. Please, today, learn about the color changes. Proteins. Proteins are the major components of your body. Everything that forms you. You are protein. Protein is you. So they serve a lot of functions. Enzymatic function, structural functions, a lot of functions. Hormonal functions. Everything in your body is just protein. Are you there? Now, the proteins you have in your body, if you don't have protein, deficiency of protein leads to a common disease called kwashiorkor. They used to ask as simple as it is. So, all of you know what kwashiorkor is. Ikungbende. So, when you break down protein, you get peptone. When you break down peptone, you get polypeptide. Abi, when you break down polypeptide, you get what? Amino acids. Is that taken? Now, from what you have here, you need to test for protein. What are the tests for protein? Burette tests, Million's tests, Santoproteic tests. These are tests you do for proteins, but please go and note the color changes. One is red, one is yellow, one is hot and no. Please, there's a code I gave them then to remember. Those that were in class when I taught this, if they still remember. But for now, I can't even start thinking about it. So please try to think about the colors of these guys. Those are the areas they most likely ask you questions. Then, where is fat here? Can I rub this guy? Thank you. No, 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 no. The structure of amino acid is CNH2, COOH, R. So that's the general structure of amino acid. Can we move on, please? So for fats, please, if you want a very good amount of energy, you need fats. But if you want fast energy, you need glucose. Highest amount of energy is given by fats. Is that taken? So if I give you a question and I say which one will give you the high amount of energy, and I put biscuits, vegetable oil, or anything that has oil, then I put eba, then I put all those things. Anything that has to do with fats, like butter, they are the ones that will give you highest form of what? Energy. Is that taking? But if you need a fast energy, it is glucose. So how do you test for fats? How do you test for fats? Osmic. Translucent. Is it the same thing as Sudan 3 test? So please, I beg you in the name of God, if you know you want to pass very well, study all these colors. The color of Sudan 3 when you add fat to it. The color of Osmic when you add fat. Translucent is just paper. Just add it to paper, then the paper becomes transparent or translucent. So that's that. Are you there? Don't worry, all these ones are the ones that used to be very long. So, minerals, vitamins. Let's quickly talk about vitamins. Now, the amounts, you only need a small quantity of vitamin, and most times get them from vegetables, from fruits, and so on. Now, these vitamins are essential in your body. They most times act as what we call coenzymes or cofactors. Just little amounts is all you need. But the question they can ask you in the exam, number one, the following fat-soluble vitamins, the ones that are not fat-soluble, that can dissolve in water, they ask you. Number two question they can ask you is that, what is the common name of the vitamin? That's number two question. Number three question, deficiency. If you don't have that vitamin, what will happen to you? Are the next questions. So we have two types of vitamins. Can I rub this part? So for vitamins, listen, oh, we have water-soluble vitamins. Water-soluble vitamins are vitamins that will dissolve in water. And it starts from vitamin B1, B2, B3, B5, B6. I'm coming. I think I still remember that code for this one. The road need plain platans for can. Thiamine, abi, riboflavin, niacin, pantothenic, pyridox, folic acid, cyanocobalamin. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7. So there's B9. Abi? So after B6, you have B9, you have B12, and you have C. So these are called water-soluble vitamins. The common name, the first one is called thiamine. Thiamine. Is that taken? That's what the word D means. Just remember the road needs plain pattern for cars. Is that taken? Now, R means riboflavin. Riboflavin. Are you there? N means niacin. P means pantothenic. This is what? I don't know the spelling again. Pyridoxine. B9. Folic acid. Is it B9 that is folic acid? Then B12 is cyanocobalamin. Cyanocobalamin. So the vitamin C, ascorbic acid. So these are the common names. Common names. Then quickly their deficiency. So they can ask you the deficiency. Deficiency is B, C, P, DAPS. That's the deficiency. BCP DAPS. They can ask you that lack of vitamin B1 will cause what? Berry. Or they will say that berry is caused by what vitamin? Do you get that? So this is berry. This is cracking of skin. This is pellagra. This is disorder of nervous system. This is anemia. This is also anemia. There are different types of anemia. This is pernicious anemia. This is scurvy. So these are your common questions. Beriberi, scurvy. Scurvy is this, I think, softening of the bones, muscles, and all. So don't forget that. Is that taken? So these are the deficiencies of, if you don't have any of these vitamins, these are going to be your problem. Vitamin B12, pernicious anemia. Can I go on? So let's look at the other types now. We have what we call fat-soluble vitamins. Can I rub the board? So for fat-soluble vitamins, don't worry, no roman stress waju. For fat-soluble vitamins, just remember headache. Oh, then remember adekanlese. Headache. Then you need to remember red cats. I don't know. I can't remember. That stands for retinol. And retinol for philo. How do you spell it? Gbogbo ẹ̀ kàn full lọ ni. PH. Okay. Ó kàn full lọ ni. So you have the phylloquinone and all of those things. So please, you need to know their deficiency. Don't forget this one. Lack of vitamin D. This one causes night blindness. They can ask you. That's lack of vitamin A. Night blindness. Ojú ẹ máa wò bye. This one causes rickets in children. Osteomalacia in adults. Tocopherol ńkọ́? Sterility. Sterile male, sterile female. Phylloquinone. Blood clotting. Thank you. So night blindness, rickets, sterility, and hemophilia. So not rating sodium. So you are not rating sodium. Water makes up 70% of your body, and water is useful in dissolving your enzymes. Water is very essential in your body. That's all. Roughages. What's the function of roughages? They are fibers. They help you to digest food. Is that taken? Then the minerals. All I would just say is we have micro elements and we have macro. Are you through with this board so I can rub it? So for elements or minerals, you have what we call trace or micro elements, Abi. Then you have what we call a trace micro or macro. Then you have macro elements. So these elements are the ones that are so essential and you require them in large quantity in your body. Then we call them macro elements. Example of macro elements are nitrogen, phosphorus, sulfur, which one again? Calcium, potassium, carbon, magnesium. Is that taken? Try to know their function. Every time you are hearing protein production, you need nitrogen. Eh? So Ross, okay. Nitrogen helps in protein formation. Phosphorus helps in HCP production, adenosine triphosphate. Magnesium, note it for plants. It is useful in the formation of chlorophyll. Magnesium is important in the formation of chlorophyll. Those are the likely ones they can ask you. Then when you want to balance electrochemical cells, you need potassium in the body. Then calcium for bones, Abi, and all. So then trace elements, you have molybdenum, boron is part of them. So all these are trace elements. They are elements you require in minute quantity. Is that taken? Can we rub the board now? Can we move on to the next thing? So we are through with nutrition. So let's quickly look at, can you solve questions on nutrition? Please follow what we have said and pick up past questions and solve. Just be careful. The thing is that you can form maybe 5 of you after class and solve it together. It will help you to discuss and it will help you to retain it more. Is that taken? Than going to sit down. But when you are forming association, don't start gisting about other things. That's why your association should not be more than 4, maximum 5. Hey, God, or Juman long group. Pair yourself like that. Then, out of the 5, pick up this nutrition now, solve everything. Oh, then anything that is causing argument, carry textbook, check it through, ask one another before you ask AI. So can we move on now? So which one are we moving on to now? Transport system. So let's quickly look at transport system. So I'm going to summarize transport system in a way that we can quickly do it. So transport system is just talking about the movement of materials within the organism, either plants or animals. Is it for you are pressing? It's phone that all of you are focusing on phone there. What are you now doing? Math students. And if you are done, Mr. Ola, go and be solving questions or you go out. You start with someone that is taking biology and not remote phone. Phone. If you have done math, please go and sit down outside, sit down somewhere at the back, be solving questions or be going home. You still have your board, practice together. What causes antigen? Oh, what causes allergy? Allergens really have antigen. What's allergens? Allergy. Allergy is a reaction, is a sudden reaction to anything that triggers it. Either a foreign agent, it can be it can be dust, it can be anything. But if I give you break now, what have we done? Mean that I want us to finish all the systems before you have break. So you did not eat. We started around 10. It's not like we started 8. Did you have any class before 10? If you know, my eyes like this is stretching. And you don't know what that means. I'm going through a lot. I'm not eating too. You see, what I just said is that biscuit. So please, we can't take break now. You have transport system, respiratory system. All these things you are doing, God self, is more stressful than class. Having to shout and shout that you should keep quiet. Maybe we should stop this class. Gbogbo let him already. You people are stressing me already. I see the people we are saying they should keep quiet. If I walk out of this place, that's the end. That's the end of this class. Because I know a lot of you have read everything. As much as I'm saying keep quiet, you will not listen. It's working. Oh yeah, bring it. Don't worry, they will not connect it. So please. It's okay. So please, it's okay. Let's stop shouting. And that's why I told you to prepare. We are going to do physics on Saturday. And today is Thursday, Abby. So please. All this talk, let's stop it. Let's stop it, please. I will send you out now. If you know it, go outside. It's simple. If you are math students, please don't sit in this place. If you know it, go outside. We have not done anything. We will still do all the systems. Do you already know transport system? I should move on to the next one. Now, every time you see questions on transport system, everybody, there's no time I've ever written JAM that I don't see question on transport system. So you should pay attention. And nutrition too. That one is very wide. So you definitely see questions. So transport system. Now, we are not wasting time. We are looking at transport system in both plants and animals. But we are focusing on everything all together quickly. I'm still going to talk about plant anatomy quickly today. So for transport system, We are talking about movement of materials within the body. That's what you say. So materials that move within the body, how do they move? What is the function of transport system? You need to know. Now, in plants, what helps them to move material is what we call the cell sap. Cell sap. It helps plants to move things within the body. Now, there are some organisms we call unicellular organisms or lower organisms, not only unicellular. These lower organisms, they move things just by diffusion. Just simply by diffusion. That's how they move things. And that diffusion, they were able to carry it out because of their large surface area. Large surface area to volume ratio. So because of all those lower organisms, they have large surface area to volume ratio. So things just diffuse in their body. They are not that big. But for someone like me now, I need blood in my toes. I need blood at the upper part of my head. So blood has to move all within my body. So for other animals, animals use blood and lymph to move material. So now, let's look at blood. Blood in animals can be separated into their components. By what we call centrifugation. So when you centrifuge blood, you are going to see a layer, 3 distinct layers. The reddish portion that is very, very thick down the layer, those are what we call the RBCs. They are the red blood cells that make up the blood. Then you have a thin layer. This RBC makes up 45% of the total blood volume of everything about the blood. Then we have what we call the plasma. Plasma makes up 55%. This one is not to 45, it's 40 to 45. There's a range we always give. But for your own, we can just give 50 or 45. Then we have what we call WBC and platelets. This one they make up 1% of the blood. So if you look at the components of the blood, we have RBC, we have WBC, and we have plasma, and we have platelets. Are you following? All of you have blood in you, so you should know what I'm saying. In some exams, they can ask you, if you want to produce blood in your body, what happened? There's what we call hematopoiesis. Hematopoiesis is formation of all of these guys, everything. But when it involves blood, it is called erythropoiesis. So formation of blood, in case they ask you, is erythropoiesis. Now, where is blood produced in the body? There are different regions depending on the of our level. We all need to know that blood is produced in the bone marrow. Is that taken? Are you following? Blood is produced in the bone marrow through a process called erythropoiesis. Now, red blood cell, you need to know this about them. Number one, they are biconcave. That's the shape of RBC. Are you following? You also need to know The red blood cell is anucleated. Anucleated means it lacks nucleus. Functions in clotting of blood. Just in a simple way, so I can move on to the next thing quickly. For platelets to help you clot blood, there is what is called thrombokinase. The thrombokinase is activated. It activates what we call thrombin. Then thrombin activates, it activates prothrombin. Then prothrombin activates thrombin. Then thrombin activates fibrinogen. So fibrinogen produces fibrin, for simplicity. So during blood clotting, you know what blood clotting is? When someone has a cut and is losing blood. I'm not taking time to explain everything because we don't have much time. When you have a cut, now, in order not to lose too much blood, that place has to be covered. So the network of mesh, we call it mesh network. That's fibrin. So there will be blood that will clot. They will coagulate together and form on the surface of that wound so that you don't lose blood. Then that is called clotting. Everything takes place inside the platelets. Is that taken? So they can ask you, it is fibrin you produce that is called that clot. So that is the fibrin. So all these ones are just enzymes that lead to it. So quickly, unable to clot. Shebi, we've talked about that. If you cannot clot blood, what do we call it? That's what we talked about in the last listing. So please, can I rub the board now? So the ability of your blood to clot now depends on all these things you have. It's a very complex process. It's a complex process. And we call this process itself hemostasis. And it's one of the things we are doing too. There are a lot of things that the spleen helps you to do. Please, let's move on first. I'm coming. Thank you. Can I rub this? Oh yeah, are we following? I'm not too fast though. Abikin Beresin Sare. Can I rub the board now? I can rub red blood cell at least. I know you should be listening and writing. And when you get to a stage, you look at the board and you're also writing. It's not every time that you'll be looking at your book. I'm waiting now. So, if you cannot clot blood, you now have a cut and your blood is not clotting. We call it hemophilia. So, your inability to clot blood. So now, let's now look at the most important thing that they always ask you in JAM. Now, we have what we call cardiovascular system. In cardiovascular system, blood is transported all around the body, but in different ways. We have some vessels that help you to carry blood. We have an organ that helps you to pump the blood. Then we have some chambers that are involved, depending on the organism you are talking about. Those vessels are your arteries, veins, and then capillaries. Now, note something. This is your heart. This is your body. This is your lungs. They can ask you this question. The pumping of blood from hearts to the body and from the body back to the hearts, we call it systemic circulation. Are you there? The pumping of blood from your heart to the body comes from your heart to the body, then from your body back to the heart. That's called systemic circulation. The pumping of blood from your heart to the lung and from the lungs back to your heart is called pulmonary circulation. All these are questions you are likely to see. That is one, based on how it is transported. Number two, I'm coming back to my trees, veins, and all. You can have blood sent from the heart without vessel into empty space. That empty space is called hemocoel. You can check the spelling well. So now, when blood moves from your heart to an empty space without vessels, then we call it open circulation. Now, because blood is going from your heart to the body, empty space, not you now. There are some organisms that exhibit that. Without a vessel, without passing through anything, then we call that blood hemolymph. Lymph is the body fluid. Hemo is the blood. So when blood goes into the body from the heart, then from the heart back to the body and not from the body back to the heart and not, then it will not pass through vessels at all. No vessels. All those arteries, veins, they don't have it. So it just passes through the body. Then we call it open circulation. Then the blood is called hemolymph because it will mix with the body fluid. Is that taken? The space where it is pumped to in the body is called hemocoel or hemocoel. So when there is vessel involved, then we call it closed circulation. So your heart pumps blood now. Something takes the blood away from your heart. That is called artery. That's a vessel. It takes it to the body. Is that taken? Then something takes it back from the body to the heart. That is called vein. So those are vessels. They are pipes through which it goes. Now, this process of sending blood from the heart to the body, from the body back to the heart is called what? What type of circulation is that? Closed circulation. Heart to the body is called what? Systemic circulation. Is that taken? So now, you know the difference between open and closed. Open is just directly into the body, no pipes. It's just like you are pouring water into your house. There's no pipe bringing the water. So, Then we have what we call single and double circulation too. I'm still on circulation. So we have single and double circulation. Single circulation means that the blood passes through your heart once. Before the blood will go into your body, it only passes through the heart once. And that is called single circulation. Double circulation, it passes through your heart twice. So look at the difference here. Blood leaves your heart, it goes to the lungs. Abi, from the lungs, where did it come back to again? The heart, before it now goes to the body. So that means that it passes through your heart. The blood that is coming enters your heart. Then when it leaves your heart, it goes to the body, which is lungs. That same blood come back again to your heart before now sent to the rest of the body again. Is that taken? That is called double circulation. Some animals, if you have done classification, you would have had that. Some fishes have 2-chamber hearts, some have 3-chamber hearts, and all of those things. So now, let's now see how this circulation goes. From what I've said here, if they ask you, what vessel help you to carry blood away from your heart? Artery. What vessel brings blood to your heart? Is that taken? Can I rub this now? I think all this makes some sense to you. When the blood passes through your heart twice, it is called double circulation. When it passes through your heart once, it is called single circulation. When blood is pumping to the body without passing through, what are the examples of organisms that exhibit open circulation? Majorly arthropods, your insects, your mollusks. Is that taken? But your earthworm and others, they exhibit closed circulation. So they don't just pump blood. So you see that black thing. All these are questions they can ask you. Can we move on? Can we move on now? Please, I want to move on to, I might still leave respiratory and excretory. I want to move to skeletal and probably nervous and hormonal. So, are we through with this? Now, can I rub you? Please, do we understand the concept? Eh? Yes. So now, let's look at the heart itself. How does blood now move in your own body? What helps you to carry your body, your blood? They are arteries, veins, and capillaries. Point of exchange is just capillaries. Now, from what we have said, arteries carry blood away from your heart. Abi, this one carry blood towards the heart. Is that taken? Most of the arteries, they carry oxygenated blood. Except which one? Except pulmonary artery. You will see how it happens now. These ones carry blood away. They carry deoxygenated blood, Abi. Those are the questions they now ask you in JAM. Veins carry blood away. They carry deoxygenated blood except pulmonary vein. Another thing is that arteries lack valve. Valves are openings. So these valves, you have some exception, except aortic valve, pulmonary. There's one tricuspid valve and all of those things. Let me leave aortic valve first. We will see when we draw it. Then these ones have valves. And they can ask you that what is the function of valves in veins? They prevent backflow of blood. They prevent backflow of blood. Please, can I get a better marker so we can? I want to rub the whole blood now. Now, let's look at the structure overview. So the structural overview, without being artistic, I want you to follow these steps through so that you will not forget. You can't study it on your own easily. So look at it. In the body, anything above the body is usually called superior. Anything below is usually called inferior. Please, I'm waiting on the marker. So your heart that is located almost behind your sternum. Your sternum is here, so your heart is here. It takes a cut with your left lungs and all of those things. So now nobody is getting marker. So when blood is coming from your lower legs, your abdomen, your intestine, coming up from your lower parts to your heart, something that carries it is called inferior vein, and we call it inferior vena cava. When it's coming from your head, from upper parts of your body to your heart, what brings it is called superior vena cava. Remember, vena, I said anything bringing blood to your heart is called vein. Abi, vein. So inferior vein or vena cava will bring it to your heart. Superior vena cava from the top will bring it to your heart. Then how will it now move in your heart is the next thing. So let's say this is the structure of the heart to start with. There is a vessel that vessel opens into a chamber. Okay. Now, in this chamber, there is another vessel that is coming from the top. So quickly, please. That vessel also opens into this place. Now, this part of your heart, this part is called auricle. But if you stand like this, this is your heart. So this is called right auricle. Now, blood comes in from the top. You know, the blood is coming from your body, Abi. The blood coming from your body, one comes from below, one comes from the top. Now, from the top, it enters this place. From below, it also enters this place. Now, this place will relax. When it relaxes, it receives the blood. Then that relaxation, we have, sorry. The type of blood that comes in from here, please, is not red, so to say. It is deoxygenated blood. It's coming from your body. So now, when it enters this place is called the right ventricle. This place that it will pass through to enter here, that place is called the tricuspid valve. Tricuspid valve. I will spell it soon. You are tired? Are you tired? No. Sit properly. You are tired? You want to go? No. If you are tired, please, let's go. Some of you are looking at, if you are tired, we can stop. But if you already know it, you can go. So that we don't waste your time. Just go and be practicing your own. So now, tricuspid valve, right ventricle. Now, there's now a channel that goes out of this place. That channel also has an opening. This channel is called pulmonary artery. Pulmonary artery. So the blood that flows in here now flows out of here. enters this place, then go out of here. I hope you are following the movements. Now, this one called the pulmonary artery. We now have the lungs. The blood flows out of here, then go to the lung. From the lungs, it picks oxygen at it can release CO2. When it picks oxygen, it becomes oxygenated blood. Oxygenated blood. Then this oxygenated blood comes back to the pulmonary artery. Comes back to a vessel. Yeah. Now, you will now know why we said one carries the oxygenated blood soon. So, this one that it comes in through is the pulmonary vein. Then it enters this place. This place that is entering, that place is your left auricle. So if you stand like this, this is your left. So this is your left auricle. Well, there is no need for any systemic structure or something. Then from here, the blood enters here now, then enters here. Then this place is called your left ventricle. Now, your left ventricle, usually the wall around here is always very thick. And your heart is protected by a membrane called pericardium. So, the wall here is thicker than the wall on this other side. Now, when the blood enters here now, then it fills this place. Then it will now enter another chamber. This chamber up. It will enter here. Then it will now move out. And go through the rest of your body. Now, this one that it enters is what we call your aorta. So now, let's analyze it again. Your body, your lower body, lower parts of your body, the upper part of your body. So I can be fast now. So blood enters your lower part of the body through what we call the inferior vena cava. Are you there? Look at them. So it enters your upper body through what we call the what? The superior vena cava. So when it enters there, where will it drop to? So from inferior vena cava or superior vena cava, it enters the right auricle. Abi? Right auricle. It's spelled this way, right auricle. In some textbooks, you see it as atrium. Atrium. So now there's a valve here where the blood will go out from. That valve is called tricuspid valve. So it will now pass through tricuspid valve and be released to where? Right ventricle. Are you following? And be released to right ventricle. There's a common question they ask you every year all around here. Then from the right ventricle is now moving through your pulmonary artery, but through a valve. That valve here is called pulmonary valve. So this valve here is called pulmonary valve. So through pulmonary valve, it goes to pulmonary artery. Then from pulmonary artery, where did it go? Are you following? It goes to the lungs. Then from the lungs, it comes back to pulmonary vein. Then from pulmonary vein, it enters where? Left auricle. Now there's a valve here that also controls the flow of the blood at the left auricle. That is called the bicuspid valve. So the bicuspid valve controls what enters the left ventricle. Are you following? Then from the left ventricle, it enters this one. That's the biggest artery in the body, the aorta. But through a valve again, that valve is from the name aortic valve. So it enters the aortic valve to the aorta. Then from the aorta, it will now go throughout the body. So that is the blood flow. So it goes from, I will call one of you to explain this now. I can see those of you playing because I'm taking my time. So inferior vena cava, superior vena cava to right auricle through tricuspid valve. Don't forget the word tricuspid as R. So right auricle and right ventricle, what separates them is tricuspid valve. What separates left is bicuspid. But the ones that has R is for right. Why the ones that? So they can ask you, why is this place thick? You know, when the blood gets here now, it will pump it. Your auricles used to relax. Auricles are meant for relaxation. Ventricles are meant for pumping. Ventricles are meant for pumping. Are you there? So the ventricles, they help you pump blood. This one will pump now to the lungs. This one will pump to the whole body. It will pump it from left ventricle to enter the whole body. So it will send to your head, to every part of your body. So now, when you associate this word, systole and diastole, systole and diastole. So systole is talking about the relaxation, while diastole is talking about the pumping. So the two of them can relax, systolic relaxation. Then the two of them can pump. It enters the stomach, it enters the kidney, it enters the small intestine, it enters the limbs, your legs, and so on. Then it goes back again to the heart. But between the liver and the small intestine, there's an association. There's a link from the small intestine to the liver. They are close together. You know, if you draw the digestive system, I just arrange them, but it does not mean that small intestine is far below. So there's a link between the liver and the small intestine. There's a portal. That portal is called hepatic portal vein. So all the ones you can see now, blood is coming from the heart to the body. Everyone that brought it from the heart to the body, they are arteries. But anytime you now hear liver, if they say that which of the vessels will carry blood, especially oxygenated blood, from the heart to the liver, then that is hepatic. So every time you hear hepatic, you are talking about liver. That's hepatic artery. For stomach, what do you call that of stomach? Mesenteric artery. For kidney, are you kidding me? Renal artery. Intestine. So you have the celiac artery. Then for the limbs, but these are the lower part of your body. If it's not sending it to the upper part of your body, it can send it to your hand, it can send it to your head, Abby, or neck. It can send it to your neck. So what are the arteries that supply your hand? Then it can also go back through that superior vena cava. What are the ones that supply your hand? Subclavian. Subclavian artery. You can see that it's simple. The ones that supply your neck, you just try to solve questions, get used to it. Just try to solve questions, get used to it. So don't forget that these ones have two portals. So it's a special part. So now for plants, how do things move in plants? Maybe I should pack that all together. Xylem is what helps to transport water in plants. I'm still going to talk about xylem and phloem because I want to teach you the plant's anatomy. Xylem, phloem, cholenchyma, parenchyma, cambium, and all of those things. I want to teach you. So let's switch. Next topic. Nervous and hormonal coordination, Abby, or skeletal system, which one? So yeah, let's do nervous system. Do we understand transport system? I told you Martin, just this morning, 1 hour. It's not even what I'm teaching you. It's not what I'm teaching you. In 1 hour, that advance, not the one for this level. In 1 hour. The blood always moves down in artery, and that's why most times veins have valves. So valves don't allow. Note this. They can ask you, can I rub the blood now? Thank you. So don't forget, inferior or superior vena cava to right auricle, to tricuspid valve, to right ventricle, through aorta, then to the lungs, then to the pulmonary vein. So you can see now, the blood that is coming from this side, from your right side now, they are deoxygenated. So deoxygenated blood are coming from here. Then when they get to the lung, they will pick oxygen and become oxygenated. Then it will now enter this place. But if you notice something the other time, I said veins always carry what? What type of blood does vein carry? Deoxygenated blood. But look at this pulmonary vein. What is it carrying? Oxygenated. So all veins carry deoxygenated blood except pulmonary vein. All arteries carry oxygenated blood except pulmonary artery. Are you there? So look at this. This is how blood flows. It goes from arteries to arterioles. Arterioles are smaller arteries. Then from arterioles to capillaries. Capillaries are the smallest, thinnest. Then from capillaries to venules. Then from venules to larger venules and veins. Now listen, blood always goes from artery to capillaries. But blood leaves from capillaries to veins. Blood goes from veins to capillaries. The blood away. I hope you get my point. When they ask you, I won't say very long. So are we done? Should we solve questions on this? Yes. Well, yeah, let's open our past questions on transport system. Everybody! Don't get tired, don't get tired. And the one that should be complaining. You don't know that when you have an exam, you should be having this energy. Yeah, where you keep them and label. You don't really know. You did not say I wish I prepared for this class very well. I wish I prepared. When you go through pulmonary artery, if they ask you to arrange it, where will you first go through? Heart. I know it's heart. Which part of the heart is in your right ventricle already? So where will you go through from your right ventricle? Of the liver, and it begins and ends with capillary. Show you the blood vessel which carried digested food from the small liver. Why is the part labeled 5 thicker than 6? You know I told you that your left ventricle has a thicker wall than the right ventricle. Why? Because it needs to pump blood. So they now ask, why is it thicker? 5 is the left ventricle and 6 the right ventricle. It's true, but does that solve your problem? It's not even the ventricle per se, it's the walls of the ventricle, the pericardium. 6 is the left ventricle, problem solved. 5 receive blood from the left auricle, is that the reason? 5 pumps blood around the body and 6 pumps blood into the lungs. So 5 have to pump blood into a very far distance, but 6 will just pump blood into the lungs, which is not too far, to the heart. Show you, that's why that one is tinier and the other one is thick. I mean here. Number 5. Which of the parts labeled is the vena cava opening? Vena cava opening. Vena is not 8. It can be 8, but from what they label, it looks more like 3. 8 is not even the option. So 2, let's check 2. 2 is what? Pulmonary artery. That's 2. 3 is what? That's superior vena cava. 4, that should be like your pulmonary vein, however it is. Then 9, you go 9 what? What's 9? Pulmonary valve. Pulmonary valve, that is 9. Do we get that? Good. So the best answer to pick here will be the win. Number 6, if the back and phloem then I'll go to skeletal then I'll go to skeletal system. So through that plants anatomy. Because there are a lot of questions you used to ask there. I'll give you a break. You go and eat. And we still have a lot to do. You people already know genetics now. Not so much in genetics. Don't worry, for Nabos and Mona, I will not waste time. We can finish those ones on time. Then, for ecology, I can move to genetics instead of ecology. I will summarize genetics to you. Oh yeah, let's go on, please. Number 6. Okay, number 6. Water salt remain the ring withdrawal or factor of food does not reach the roots. The roots got to be deep. Food will not get to the roots. Number 7. They've answered that before. Number 8. The part matrix is what? Pulmonary artery. Which of the following is not a function of mammalian blood? Let's look at it. Engulfing bacteria, phagocytosis, Sabi? I told you about white blood cells, right? Number 2, production of hormones. Is it blood that produces hormones? No. Blood only carry hormones. We are going to see that in the next lesson. Transporting urea, yes. They help to transport waste materials to the kidney. If we are done excretion, you see that. Then distribution of it, yes, through vasoconstriction and vasodilation. Vasoconstriction is constriction of the blood vessels to resist the outflow of blood, while dilation is opening of the blood vessels and allowing the blood to flow very well. For the distribution of heat. That one is just a special pattern. Abi, that will supply the three blood vessels that supply the mammalian and electric canals. You have the stomach now, it's part of the alimentary canal. What supplies that? The mesentery. Then you have the small intestine, which is part of it, that's celiac. So, which one is segmented artery there? Hepatic will go from small intestine to liver. I think the best answer is it. Because celiac is there. So, and eccentric is there. So that's so you can confirm that later. But that's the answer. Number 11. Which of